In a previous technical guide video, I introduced some basics of the colony-forming cell or colony-forming unit assay. This is Ivan Rich, CEO of Hemogenics, and I shall be talking about and demonstrating the Methylcellulose ColonyGro reagents and the miniaturized colony-forming assay kit CAMEO-4. ColonyGro is a methylcellulose colony-forming reagent, similar to other commercially available methylcellulose reagents, such as Methylcellulose. However, ColonyGro has a different formulation that makes it easier to use and allows a larger number of cultures to be produced from a 100 ml bottle of reagent. In fact, more than 40 cultures can be performed. CAMEO-4 is a miniaturized colony-forming assay, but uses exactly the same reagents as ColonyGro. CAMEO-4 requires a total volume of only 0.6 milliliters, including the cells. The CAMEO-4 assay kit also contains 50 4-well culture plates. This saves ordering additional supplies. Each well will contain 0.1 milliliters of the CAMEO-4 culture master mix. CAMEO-4 requires less time to set up, less time to count colonies, and is very cost effective. The procedure for performing a colony-forming cell assay is essentially the same using ColonyGro or the CAMEO-4 assay kit. Larger volumes are used for ColonyGro. Because CAMEO-4 is a microassay, greater accuracy in dispensing the reagents is required. In the lower picture, the contents of the CAMEO-4 kit are displayed. They include the CAMEO-4 master mix for a specific cell population, base medium to dilute the target cell suspension, and 50 sterile 4-well 35 mm Petri dishes. Naturally, an assay manual is also included. Hemogenics has developed ColonyGro and CAMEO-4 to detect up to 20 different cell populations from the lympho-hematopoietic system derived from human, non-human primate, horse, sheep, pig, dog, rat, and mouse tissues. These include 8 different stem cell populations, 9 different progenitor cell populations, and 3 precursor populations. 5 of these populations are equivalent to those detected using human Methylcellulose reagents. In addition, you can also obtain ColonyGro and CAMEO-4 without any growth factor cytokine cocktails. This gives you the flexibility to add your own, or use this formulation as a control. To dispense either the ColonyGro reagents or the CAMEO-4 master mix, use an electronic positive displacement syringe pipette, such as that shown in the picture. This particular model is an Eppendorf stream. For dispensing the ColonyGro reagent, use a sterile 5 ml syringe attached to the pipette. For dispensing the CAMEO-4 master mix, use a 1 ml or 2.5 ml syringe. Although electronic pipettes still need to be professionally calibrated at least once a year, electronic pipettes calibrate themselves internally every time they are used. Please do not use syringes and needles. These are responsible in part for the high variations seen with competitors' products. Prepare the cell suspension in the normal manner. It is best to remove red blood cells by lysis or density gradient centrifugation, since high concentrations of red blood cells can inhibit colony growth and make it very difficult to identify the colonies. Please note that usually a 10-fold greater cell concentration is needed for the macro ColonyGro assay than CAMEO-4. In this case, we are starting out with a suspension at 500,000 cells per milliliter and will end with a final cell concentration of 5,000 cells per well. For both ColonyGro and CAMEO-4, the working cell concentration is added to the master mix at 1/10 of the total volume. For CAMEO-4, the total volume prepared will be 0.6 milliliters. To each of the 4 tubes in the picture, 0.54 milliliters of the CAMEO-4 master mix has been added using a positive displacement pipette. Now 0.06 milliliters of the working cell concentration at 500,000 cells per milliliter is being added to tube number 1. This will dilute the cell concentration tenfold down to 50,000 cells per milliliter. Once the cells have been added to the master mix, this is now the culture master mix. Cap each tube and use a vortex to mix the contents. Here we have already plated 4 wells of the first sample in tube number 1, and we are now in the process of dispensing 0.1 milliliters from tube 2 into each of the 4 replicate wells. Dispensing 0.1 milliliters into each well further reduces the cell concentration another tenfold, down to the final concentration of 5,000 cells per well. Use a 0.5 or 1 milliliter syringe attached to the positive displacement pipette to perform this step accurately. When 0.1 milliliters of the culture master mix has been dispensed into the well, the reagent will usually spread out by itself to cover the whole surface. If this does not occur, place the lid on the dish, lift the dish at an angle and gently swirl the plate until the contents cover the surface of each well. As with other colony forming assay procedures, it is best to use a humidity chamber to prevent the cultures from drying out. This is despite the use of a fully humidified incubator. To do this, use a 100 mm sterile Petri dish. Place 2 35 mm Petri dishes in the large dish, together with one 35 mm Petri dish filled with sterile water but without its lid. Transfer all the cultures to a 37 degrees Celsius humidified incubator with an atmosphere of 5% CO2 and, if possible, 5% O2. Using low oxygen tension increases plating efficiency by decreasing free oxygen radical formation that occurs at normal atmospheric oxygen tensions of 21% oxygen. Hemagenics is the only company that has made advances to the colony-forming cell assay. That advancement comes in the form of the CAMEO-96 assay, the first and only methylcellulose colony-forming cell assay that can be used to measure both proliferation and differentiation, and the only assay that allows the colony-forming assay to be truly standardized. Please contact us with any questions you might have. We look forward to hearing from you. Thank you.